Hello everyone, I'm going to present about a large-scale genomic identification of CAZymes families involving mannan and xylan degradation, done at the Laboratory of Computational, Evolutionary and Systems Biology at Sena USP. In a scenario of growing energy demands, the use of renewable energy from biomass can be one of the resources. However, biomass needs pretreatments to release the energy stored in the compounds, like biological treatments that can apply the use of CAZymes. And one of the CAZymes activities is the deconstruction of polysaccharides, highlighted for this activity G8 and CE classes. And most of CAZymes information is summarized in the case database that will be used further on this work. Two important polysaccharides in monocot cell walls are mannan and xylan that have a recognizable molecular structure. Thus, there is interest in finding novel CAZymes that can break them into mono or oligosaccharides. Our objectives were to determine clusters of CAZymes proteins with greater potential to deconstruct mannan and or xylan that have not been functionally or structurally exploited, performing a filtering of number of sequences predicted as CAZymes into less sequences, but with higher potential to act on desired carbohydrates, possibly uncovered functioning and structured differently. To reach this, we had two main questions: how to group sequences, how to identify not studied sequences or clusters, and how to relate sequences to mannan or xylan. Before examining those questions, we gathered genome sequences from bacteria, fungi, and archaea groups deposited on NCBI, and from this data, identify the CAZymes sequences. And to start relating CAZymes to mannan and xylan, we identified families with at least one mannanase or xylanase and worked only with sequences from these families. From these steps, we've recognized 22 families of interest for xylan and 15 for mannan, whose sequences were unevenly distributed, where the five top families in sequence amount had 73 and 84% of the total sequences for xylan and mannan, respectively. Those proteins were most identified in bacteria and for some families in fungi. Then we start answering the questions. Firstly, to identify not studied sequences, we use CAZy because once most studied CAZymes are there, every sequence predicted previously that wasn't on CAZy can be considered as not studied. Secondly, we grouped the sequences using phylogenetic inferences and hierarchical clustering, based on the relation between sequence, structure, and function. From this, we were able to represent the considerable lack of protein characterization in face of nucleotide or predicted sequence amounts. To most of the families, more than 50% of the cluster generated didn't have any studied sequences, and that means that they didn't have any information to perform inferences. Third, to relate sequences and as a consequence clusters and clades to mannan or xylan, we searched ADC codes related to mannan or xylan bonds degradation. Then we identified the sequences with the ADC codes from the information extracted from CAZy and from the results of its code deep learning prediction. And to conclude, we tagged a cluster as an interest cluster if they had at least one predicted or studied code related to mannan or xylan. With this code prediction, the total number of clusters with information becomes over twice as the original size, and the number of interest clusters compared to the initial amount reduces to more than a half, but it still comprehends a large number of sequences. And finally, the phylogeny is represented, where the outline color means the group where the sequence came from, and the leaves with black mark represents a potential xylanase, and with a light blue mark represents a possible mannanase. To conclude, the reduction in sequence amount and increase in the relation between the sequences and the theoretical possibility to deconstruct mannan or xylan were performed. However, there are still a considerable number of sequences, especially because the big difference between the quantity of predicted and characterized sequences that complicate the inferences. Thank you for your attention. More information can be found on Project's GitHub and in the laboratory site.